You're watching an animal perform its daily routine, but it's no longer in control. Across more than 200 species, hidden parasites hijack their hosts' minds, quietly overriding fear, pain, even basic survival instincts. The result? Living creatures turned into biological weapons, serving another's commands. How does this invisible invasion happen? And could it be unfolding right in front of you without anyone noticing? A fish glides through shallow water, its movements smooth and unremarkable. In the grass, an ant follows its trail, antennae twitching as it searches for food. These animals look perfectly healthy, alert, active, blending into their world. But beneath that surface, something else is at work. Field biologists have documented more than 200 species where outwardly normal animals are no longer making their own choices. Their bodies remain strong, their senses sharp, yet the decisions guiding them are no longer their own. Infected fish continue to swim, feed, and school alongside their peers, but they spend more time near the surface, exposing themselves to predators. Ants carry on with their colony tasks, but at certain moments, they break from the group and wander with strange intent. Even rodents, seemingly cautious, show a sudden disregard for danger, approaching the scent of predators they once feared. To the casual observer, nothing seems amiss. The fur is glossy, the eyes are bright, the behaviors fit the script of daily survival. But the instincts that once kept these animals safe have been quietly rewritten. The parasite's influence is subtle, hidden behind the mask of normalcy. In the wild, the line between autonomy and control is thinner than it appears. What looks like health may be something else entirely. A living creature, animated by another's will. Hidden inside these animals, parasites wield a precise set of biochemical tools. They do not simply damage nerves or flood the body with toxins. Instead, they manipulate the very signals that govern fear, pain, and movement. Some parasites alter levels of neurotransmitters like dopamine, rewiring the host's sense of reward and risk. Infected rodents, for example, show changes in dopamine signaling that shift their response to predators. In insects, parasites may target octopamine or serotonin pathways, adjusting how the host responds to light, sound, or the presence of others. Beyond neurotransmitters, some parasites can change which genes are active in the brain. In ants, fungal invaders disrupt the normal circadian rhythm by altering the expression of clock genes, causing the insect to move and bite at times that favor the parasite. Others produce molecules that suppress pain or anxiety, allowing the host to enter dangerous situations without hesitation. These manipulations are not random. They are targeted, often leaving the host's basic functions intact while silencing the instincts that would keep it safe. The result is an animal that appears healthy and alert, but whose decisions no longer serve its own survival. The parasite's toolkit, chemical messengers, gene regulators, and immune suppressors, turns the host into a living delivery system, obedient to a foreign command. Each pathway hijacked is a thread pulled from the fabric of instinct, unraveling autonomy molecule by molecule. At the heart of the rainforest, a single ant climbs higher than it ever would on its own. The ascent is not random. Ophiocordyceps, the so-called zombie ant fungus, releases waves of secreted proteins and toxins that hijack the ant's internal clock. Studies led by Dr. David Hughes reveal that infected ants leave their trails at midday, compelled by a fungal rhythm that overrides their own. The ant's jaws clamp onto a leaf vein exactly 25 centimeters above the forest floor. a height that offers the perfect blend of humidity and airflow for the fungus to grow and spread spores. The death grip is no accident. Fungal effectors infiltrate the mandible muscles, locking them tight as the ant's brain remains eerily intact. Contrast this precision with the chaos inside a rodent infected by Toxoplasma gondii. Here, the parasite triggers a 14% rise in dopamine, a chemical that warps the rodent's sense of fear and reward. Dr. Jana Lafferty's research shows infected mice and rats ignore the scent of cats, sometimes even seeking it out. The parasite doesn't orchestrate a single fatal act. It dials up risk-taking across the board, turning caution into boldness. In both cases, the host survives just long enough to serve the parasite's purpose, whether that means a perfect bite at noon or a reckless sprint toward danger. A cricket stumbles through damp grass, its body carrying a silent passenger. Inside, a hairworm coils and waits. When the time is right, the parasite releases a suite of chemicals that alter the cricket's sense of direction and thirst. What once was a healthy fear of water is replaced by an irresistible urge. The cricket veers toward a puddle or stream, sometimes at dusk, when its normal instincts would keep it hidden. Without warning, it plunges in. The hairworm bursts free, writhing into open water, leaving the host behind. Elsewhere, a tiny ant clamps its jaws onto the tip of a grass blade as night falls. The ant remains motionless, exposed, exactly where grazing mammals are likely to feed at dawn. This is the work of the lancet liver fluke. The parasite's life cycle depends on the ant being eaten by a cow or sheep, so it manipulates dusk behavior, switching the ant between normal activity and this lethal grip. Through subtle changes in neurochemistry, possibly peptides and other modulators, the parasite turns the host into a living vehicle, delivering itself to its next environment. The animal remains alive, but its body and movements are no longer its own. A snail clings to a leaf, its eye stalks swollen and striped in unnatural greens and browns. Inside, the trematode Leucochloridium paradoxum pulses in sync with the snail's heart, the brood sac throbbing up to 70 times per minute. This living lure flashes like a beacon, mimicking the caterpillars that birds instinctively hunt. Drawn by the color and rhythm, a songbird strikes, tearing off the eye stalk and swallowing the parasite in a single, violent motion. The snail, blinded but alive, may regrow its stalk and face the cycle again. Elsewhere, a frog drags itself through shallow water. Its hind leg splits into a twisted branch of flesh, an extra limb useless for escape. Infected with Ribeiroia trematodes during its larval stage, the frog's development has been warped from within. Birds like herons and egrets gather at these ponds, snapping up the malformed prey with ease. Field studies in California wetlands show that predation rates on deformed frogs can double or triple, transforming the host into a delivery system for the parasite's next generation. Each grotesque form, each pulsing lure, is a calculated adaptation, an animal's body turned against itself, its survival rewritten as a weapon for another's gain. Once a parasite has seized control, escape is almost impossible. The host's immune system, built to recognize and destroy foreign invaders, is left searching for threats it cannot see. Many parasites survive by cloaking themselves in molecules that mimic the host's own proteins. This molecular disguise lets them slip past immune patrols, hiding in plain sight. In some cases, knockout experiments show that when these mimicry proteins are removed, the host's defenses can finally respond. Yet most parasites never give the immune system that chance. Their effectors blend seamlessly with the host's tissues, dampening inflammation and steering immune cells away from the sites of manipulation. Even as the host's body remains alive and functional, the machinery of self-defense is quietly bypassed. Across worms, fungi, protozoa, and wasps, this ability to remain undetected has evolved many times, each lineage finding its own way to silence alarms. For the infected animal, reversal is rare. Once the takeover begins, the path back to autonomy is almost always closed. Rabies spreads through a bite, but its true weapon is fearlessness. Once inside a mammal's body, the virus travels along nerves to the brain, concentrating in regions like the amygdala, centers that govern aggression and panic. The result is a living engine of violence. Animals that once avoided conflict now lash out, biting anything within reach. Each attack is a chance for the virus to move on, its genetic code riding in a spray of saliva. Infected raccoons, foxes, and bats become vectors of chaos, reshaping the risks for every animal and human in their territory. The ecological impact of such manipulation ripples outward. Studies of California estuaries show that when parasites drive fish to the surface, predation rates can skyrocket by 10 to 30 times. Food web models reveal that these altered behaviors don't just affect one species, they redistribute energy and change who eats whom across entire habitats. Now, biotechnology researchers are studying these natural strategies. Programs like DARPA's Insect Allies aim to use insects as living delivery systems for engineered viruses, echoing the tactics of parasites. Critics warn that harnessing such power blurs the line between defense and weapon, raising questions about control, intent, and the unforeseen consequences of rewriting behavior at the molecular level. In nature and in the lab, the ability to turn living creatures into tools is both a marvel and a warning. Right now, hundreds of parasite species are shaping animal behavior in ways we're only beginning to understand. The line between autonomy and control in nature is never fixed. As science uncovers more of these hidden manipulations, we're forced to ask, how much of free will is truly our own?